Hello, my name is Haneen Hamed. I am a postgraduate student in King Abdulaziz University in Jeddah, Saudi Arabia. This research is for the requirements of a master's degree in biology and genetics. Our topic is about investigating the effect of cigarette smoking on the NKX3.1 and TMPRSS2 genes associated with male fertility. Here is the outline of our presentation. We will talk about introduction, the aim of study, methodology, results and discussion, and finally conclusion and references. Male infertility is defined by the failure to achieve a clinical pregnancy after 12 months or more of regular unprotected sexual intercourse. There's a wide and interacted causes of male infertility. They include medical causes and environmental causes like industrial chemicals, and lifestyle and health causes like tobacco smoking or being overweight or alcohol use. Smoking induces oxidative stress and thereby affecting sperm viability and morphology and function, thus reducing male fertility. However, findings on the impact of smoking on male fertility are not consistently conclusive. In many previous studies, single nucleotide polymorphism are used as molecular markers in studying diseases. This can be done by case control studies, including large-scale SNP genotyping in a patient group and healthy control group to compare differences in genotype for the studied diseases. Then the relationship between a specific genotype and the phenotype is used to characterize susceptibility genes that are associated with the disease. This pioneering study in Saudi Arabia aims to provide baseline information for future research. This figure shows the effects of smoking on male infertility. Smoking can affect sperm structure and proteins and can raise oxidative stress and can affect semen parameters and seminal plasma and sex gland function, and also may cause chromosomal damage. Our aim of this study is to investigate the impact of cigarette smoking on NKX3.1 and TMPRSS2 gene, SNPs, and their implications for male fertility. Our selected targeted genes for SNP analysis are first the NKX3.1 or NK3 homeobox 1 is located in chromosome 8. It's a homeobox gene primarily expressed in the prostate gland and plays a crucial role in prostate development and function. The second gene is TMPRSS2 or transmembrane serine protease 2 is located on chromosome 21, is another gene predominantly expressed in prostate epithelial cells. It plays a role in the processing of proteins involved in cellular signaling pathways. Methodology. In this study, we have collected samples into 3 groups. The first group is 25 infertile men. They have fertility problems, but they do not smoke. And the second group also have 25 smokers, all men, smokers, which they are smokers, but they don't have apparent fertility problems. And the last group is 25 control men, which are non-smokers and has no fertility problems. And we have obtained ethical approval according to the guidelines of the Ethical Committee of King Abdulaziz University Hospital, and all patients provided written informed consent for the collection of samples and subsequent analysis. Participants. The 75 men between the ages of 19 and 60 years were selected based on their health status from the Health Service Administration Department at King Abdulaziz University and the Health Plus Fertility and Women Health Center in Jeddah. Sample collection and criteria. Sample collection were based on fertility history, duration of infertility, number of children, and and semen analysis showing parameters below normal. Blood samples were obtained from King Abdulaziz University Medical Services Center and Health Plus Fertility and Women's Health Center in Jeddah, Saudi Arabia. And DNA analysis, the DNA extraction were conducted at the Center of Excellence in Genomic Medicine Research at King Fahd Medical Research Center in Jeddah, Saudi Arabia, and genotyping was done in Haven Scientific Innovation Center at King Abdullah University of Science and Technology, KAUST, in Jeddah, Saudi Arabia. In DNA extraction, we used Kiam DNA mini and minikit and blood minikit used for DNA purification of total DNA for PCR. The purity of DNA samples was determined using thermoscientific nanodrop spectrophotometer. And in genotyping, the regions of interest within the NKX3.1 and TMPRSS2 genes were amplified using real-time technique. Hormonal analysis, serum from whole blood samples were obtained, and testosterone and prolactin and luteinizing hormone and follicle-stimulating hormone tests were performed by using an LTI immunoassay system. In the results, we used statistical analysis was derived by Python, and numeric variables are described by mean, standard deviation, and median, and compared using ANOVA and t-test if parametric assumptions met, or Kruskal, Wallace, and Mann-Whitney tests in case of violation of parametric assumptions. Moreover, categorical variables are described by count, by percentage, and is compared by a chi-square test if expected count in any cell is greater than 5. Otherwise, Fisher exact test was used. P-value less than 0.05 considered significant. Here is a description of the NKX3.1 SNP and TMPRSS2 SNP of the whole samples. In NKX3.1 RS2228013 SNP, 92% of the samples have the GG genotype, which is the normal type, and only 5% of samples have the hetero SNP type, and there was 2 samples that were undetermined. In TMPRSS2 gene, RS1232970 SNP, 60% of samples were normal type, and 37% of samples were hetero and have the SNP, and only 2 samples were TT homozygous genotype. Table 2 shows the distribution of NKX3.1 SNP types among infertile smokers and control groups. It deserves noting that the hetero type did not appear in the smokers group, and it appears only 2 times in control and infertile group. Table 3, comparing different population characteristics among TMPRSS2 SNP. Basic characteristics of population under study was compared among different RS1232970 SNP, and it was clear from the table that different SNPs were not statistically significant among population characteristics. But for example, in age characteristic, although the p-value is not less than 0.05, the normal genotype percentage was higher than the hetero and homozygous types. In table 4 shows the distribution of TMPRSS2 SNP types among groups. Moreover, it was clear that around 60% of the groups have the normal type CC. This percentage is a bit more in the S group, and also CT constitutes the rest of the groups. Only the control group has the homotype TT, and it was only 2 observations. The comparison of distribution of different SNP type among each group showed a non-significant result. when comparing hormone levels among different TMPRSS2 SNP types, the TMPRSS2 SNP types were no significant difference among groups, but the testosterone was a bit lower in the CT group. Thank you for staying engaged as we explored the core results of our topic today. We will now transition into the discussion portion of our presentation. We are poised to delve into the announced findings and their broader implications, particularly concerning nicotine impact on sperm health and the role of SNPs in the TMPRSS2 and NKX3.1 genes, and our study's contribution to the ongoing conversation about male infertility. We have reviewed compelling evidence suggesting nicotine and smoking worsen oxidative stress, leading to significant distribution in spermatogenesis and DNA integrity and overall sperm function. These insights are critical considering the widespread prevalence of smoking and its potential as a modifiable risk factor in male fertility issues. Regarding the TMPRSS2 gene genotyping, the presence of RS8134-378 SNP might have implications on the expression and/or formation levels of TMPRSS2 fusion because both have been shown to be influenced by androgen. These results are not in line with our results, as ours revealed no correlation between TMPRSS2 SNPs and infertility. And regarding TMPRSS2 and hormones, no significant differences were found in hormones levels or SNP ratios of the TMPRSS2 gene, indicating no correlation between TMPRSS2 SNPs and infertility. Regarding to NKX3.1 gene genotyping, the results of the current study showed no significant differences in hormone levels between the individuals of the two different SNPs of NKX3.1, meaning that genetic variation of this gene did not affect the hormone levels in males. And regarding NKX3.1 gene and hormones, our results show no linkage of NKX3.1 gene to be NKX3.1 gene to hormonal regulation, smoking effect, or infertility. The results of the current study are not in line with the findings of Boller et al. That they found that C3T polymorphism at nucleotide 154 coded for a variant protein with a substitution of cysteine for arginine and at amino acid 52 of NKX3.1 gene. In conclusion, smoking is considered a major factor leading to male infertility. In this study, we focus on understanding the effect of cigarette smoking on the NKX3.1 and TMPRSS2 genes associated with male fertility in Saudi Arabia. Our results showed no linkage of NKX3.1 TMPRSS2 genes, SNPs, to hormonal regulation or smoking effect or infertility. This study can have important implications for understanding the effects of smoking on infertility in Saudi Arabia. Thank you so much for listening.